Good morning. Good morning, everyone. Happy Wednesday. Good morning. Good to see you. Yeah, go ahead and type your name into chat for me. Okay. All right. Well, today in lab, we're going to be talking about our last biochemical test. Or tests. We're going to talk about the Oxidase Test, and we're going to talk about the nitrate reduction test. And that will finish up our discussion of biochemical testing. So we started back with carbohydrate fermentation. We talked about protein catabolism. Well, we talked about fermentation and OF media. We talked about protein catabolism and amino acid catabolism. And today we're going to finish up talking about respiration, basically, and with a focus on anaerobic respiration. So yeah, it looks like we're still missing a few people, but we'll go ahead and jump in. The first thing I want to do this morning is just pull up our remaining schedule. That's what I'm going to do right now. That's what you should see on your screen. It's our course modules page. So this is the module for this week. Of course, you have your lecture exam, your fourth lecture exam. That's due by midnight tonight. If you haven't done it already, several of you have already done it. Good job. You've got your lecture quiz for the immune system to do. And we talked about protein catabolism on Monday, and we're doing respiration today. You actually have two sets of lab questions. I just separated them out into the two topics: protein catabolism and respiration, due by midnight on the 24th. Now, looking ahead to next week is our last week of class before the final exam. It is a busy week. So this is the second time this summer that I'm going to urge you to get started on the lecture material early, if you can. If you have some time towards the end of the week, you might want to jump in and get started on this lecture material. It looks like we have three topics here, but it works out to about the length of two topics. The intro to vaccination and the intro to epidemiology topics are both short. So together, they really constitute about one lecture topic in length. And then the other one is called pathogens and how they spread. So these are the final lecture topics for the course. And of course, we have for laboratory, we're finished talking about tests. So what we have is for next week is on Monday, we're going to be talking about your final lab assignment. And you'll see that there's a page here called Lab Materials for the Unknown Microbe Assignment. If I click on that, you'll see there's several documents. And again, we're going to talk about this on Monday in lab, the instructions, the data that you're going to be using for the assignment, and some tools that you're going to be provided to do the assignment. We will talk about that when we get together on Monday. So next week, you'll be working on this final lab project for me. Here's the link where you'll submit it. You'll be submitting the unknown assignment there. It's a decent assignment in that it's worth 50 points. So it's worth the same as a lab practical exam. So it's worth your time and effort to do the best work you can on that unknown microbe. So you'll be working on that next week. You also have your last lab practical exam next week. So on Wednesday, we don't even meet for lab. We'll meet on Monday. We'll talk about the unknown assignment. And then you'll also have your lab practical to do, but we won't meet on Wednesday. All right, it's a busy week. And then of course we have the final exam coming up. So because we don't have lab exercises to do next week, we will have an opportunity to meet or to talk about the final exam and how you should be studying for it and so on. We can do all of that next week when we get together. But like I said, if you have the chance to get ahead on your lecture material, that would be my recommendation. Get the lecture material viewed, get a good set of notes taken, and then next week you can really focus more on the lab work. So you're not struggling to get everything done. And that should leave you time then to prepare for the final exam. Okay. All right. So I'm going to go ahead and pull up our slides for today. That's what you should be looking at right now. The title slide says Lab Respiration. Now, just to review, this word oxidative, when we talk about oxidative metabolism or oxidative catabolism, remember it has nothing to do with oxygen being present or absent. Oxidative refers to oxidation reduction. The oxidative side of it refers to the loss of electrons. When a molecule is broken down, when the covalent bonds in the molecule are broken apart, that's what we call catabolism. When we build a molecule, that's what we refer to as an anabolic reaction. But breaking apart something, that's catabolic. And it's oxidative if it uses oxidation reduction reactions. And all an oxidation reduction reaction is, and remember, they always occur in pairs, either the loss or the gain of electrons. Remember, when we talk about oxidation reduction reactions, most of the reactions that you're dealing with in a living cell are this type. Most of the reactions involve, again, breaking down molecules and building up molecules. Oxidation is when the bond is broken and electrons are released. In other words, the atom in the molecule is losing electrons. Reduction is the opposite. It's the gain of electrons. So oxidative has nothing to do with oxygen, all right? It's about the chemistry that we're looking at in the reaction. Remember too, another review point, microbes have amazingly diverse metabolic capabilities. To make their ATP, some of them do cellular respiration, followed by oxidative phosphorylation. Again, oxidative meaning it has oxidation reduction reactions in it. Oxidative phosphorylation is a great way to make ATP. Cellular respiration itself can be aerobic or anaerobic. And both kinds, both aerobic and anaerobic cellular respiration, are followed by oxidative phosphorylation. Let me say that again, because again, it can be confusing where oxygen is involved and where it isn't. If you're a microbe and you do anaerobic cellular respiration, all that means is you're doing cellular respiration, glycolysis, pyruvate processing, the Krebs cycle, electron transport. You're doing the exact same thing that aerobic organisms do. But at the bottom of your electron transport chain, there is no oxygen there. There's another molecule, something like nitrate or sulfate or carbonate. If you do anaerobic cellular respiration, you just have a different final electron acceptor. And once that final electron acceptor receives the electrons that came out of glucose, you go on and do oxidative phosphorylation. Remember, that process takes the energy that was harvested in the electron transport chain and uses it to build a gradient, a hydrogen ion gradient. Hydrogen ions get pumped out of the cell and into that space between the cell membrane and the cell wall. That space that we call the periplasmic space. A gradient is built. Remember, if you want to build a gradient as a cell, you have to spend some energy to do that. That energy comes from the electrons that were released when glucose was broken down. The energy is harvested from the electrons in the electron transport chain, and then it's used. It's used to fuel the pumps that are going to make this hydrogen ion gradient. Once the gradient is built, it's allowed to resolve. It's allowed to go away. And the way it resolves is by those hydrogen ions flowing back into the cell through that very important molecule called ATP synthase. ATP synthase is a big complex protein that sits in the cell membrane. And it literally has a tunnel in a part of it. The hydrogen ions that were pumped out of the cell will flow right back in, but they're going to flow in through the ATP synthase tunnel. And that motion, that kinetic motion, is going to cause the ATP synthase molecule, part of it anyway, to spin. And it's that spinning motion that builds ATP. So it doesn't matter if you're an aerobe or an anaerobe. You're doing the exact same process with one exception. And that one exception is what molecule you're using as your final electron acceptor. So don't fall into the trap of thinking that anaerobic organisms can't do oxidative catabolism, because they can. Some of them can. Remember, there are some microbes that can do both. There are some microbes that can do aerobic cellular respiration and anaerobic cellular respiration. Some of them can even do fermentation as well. They're very diverse. They're much more flexible metabolically than you and I are. Fermentation is a different process from anaerobic cellular respiration. Fermentation also makes ATP, but it does it using a different mechanism. Oxidative phosphorylation doesn't happen in fermentation. Substrate level phosphorylation occurs. And remember, this way of making ATP is inefficient. And by that, we mean you don't get very much ATP per glucose. In oxidative phosphorylation, you get a lot of ATP per glucose. Okay, Some microbes do one or the other of these. Some microbes do fermentation, and that's it. Some microbes can do all three. It depends on the microbe. One other really important thing to note, and I wrote this in comments on your some of your quiz, the quizzes you submitted for carbohydrate catabolism. Remember, living cells use carbohydrate to make their ATP. That's what they use to make ATP. Ultimately, it's glucose. But some microbes are able to use other sugars and break them down to glucose and then use glucose. But it's all at the bottom of it, it's glucose. Glucose is the granddaddy of ATP production. It's carbohydrate that they use. Cells do not use protein to make ATP. They don't use lipid to make ATP. They use carbohydrate to use ATP, to make ATP. They just make it through different mechanisms. All right. Very important to remember that. That's not just bacterial cells either. That's all cells. All cells. Bacteria, archaea, eukarya. Carbohydrate is the molecule that's used to make ATP. All right, enough of a review. Let's look at cellular respiration with a little bit of a different view now. Let's think about where, we know where oxidation is happening. Glucose gets oxidized, glucose gets broken down or catabolized. Remember, anytime something gets oxidized, something else has to get reduced. Those two things go together in nature. You can't have one without the other. If you look at something like glycolysis, remember glycolysis is a 10-step reaction. What happens is glucose gets oxidized, then reduced. Then the next molecule gets reduced. Then the next molecule gets oxidized. Then the next molecule gets reduced. Then there's an oxidation, then there's a reduction. Then there's an oxidation, then there's a reduction. There's a whole series of them. Since we start with an oxidation, we have to end with a reduction. So oxidation, reduction, oxidation, reduction, oxidation, reduction, all the way through glycolysis, all the way through pyruvate processing, all the way through the Krebs cycle, all the way through the electron transport chain. Oxidation, reduction, oxidation, reduction, oxidation, reduction. The very final reaction has to be a reduction because we started with an oxidation. And the final reduction is the reduction of that final electron acceptor molecule. The final electron acceptor is going to accept electrons from the electron transport chain. It's going to take them in. So that molecule gets reduced. Gain electrons, reduction. If you're an aerobe, it's oxygen that's at the bottom of the electron transport chain. It's going to gain electrons, it's going to get reduced. If you're an anaerobe, it's something else. It's nitrate or maybe sulfate or maybe carbonate or maybe something else that's going to gain the electrons and get reduced. Aerobes use oxygen as their final electron acceptor. Oxygen is available at the end of the electron transport chain, and oxygen gets reduced. It gains the electrons and it gets reduced. There are many, many oxidation reduction steps in cellular respiration, but it all begins with an oxidation and it ends with a reduction. Now, the formula for that final reduction you could refer to it as the pathway of that final reduction. The oxygen at the end of the electron transport chain is going to combine with the electrons that were removed from glucose and a little bit of hydrogen ion, and we're going to create water. Now, this equation isn't balanced. None of the equations I'm going to show you today are balanced. That's not what's important for us to know. I want you to understand that this final electron acceptor is going to gain electrons, and it's going to become something else, water in the case of aerobes. This is the equation for, or the pathway of reduction in an aerobic organism that's doing cellular respiration. The electrons that came out of that glucose molecule, as each of the bonds was broken, electrons were released, because that's all a covalent bond is. It's a pair of electrons that are being shared between two atoms. When the bond is broken, the electrons get released. Those little electron carrier molecules take the electrons over to the electron transport chain and send them down the electron transport chain in order to harness their energy. Remember, in lecture, we talked about how the electron transport chain can be imagined as a staircase. And when the electrons come into the top of the staircase, they're full of energy. And as they step down the staircase, bits of their energy are removed until when they get to the bottom of the staircase, they're at a lower energy state than when they started. So as the electrons come down the electron transport chain, energy is being removed from them. It's being harvested from them. That's what the electron transport chain does. We also talked in lecture about how the electron transport chain is made primarily of protein. Remember, a bunch of proteins that are joined together in these complexes. And then there are these secondary molecules that help move things between the complexes. We talked about one of those molecules called ubiquinone, also known as Q or coenzyme Q. Q takes electrons from one place in the chain and transfers them to the next place in the chain. Well, if you remember, there was also a second molecule that does that work, and that molecule was called cytochrome. Cytochrome. There are many different versions of cytochromes. And depending on the type of organism you're looking at, whether it's a bacteria or an archaea or a eukarya, they have different cytochromes. But these cytochromes are all doing the same type of job. They're collecting electrons from one protein in the electron transport chain and handing them over to the next one. So in order for an electron to go from one step on the electron transport chain to the next step, it needs a little help. And the help comes from either ubiquinone or from one of these cytochromes. The electron is sort of stepped down, and it needs the assistance of ubiquinone or cytochrome in order to take the step. So there are lots of these cytochromes. In bacteria, there are four classes, four families of cytochromes, and they're named A, B, C, and D. They all do the same work in the electron transport chain, but they're structurally a little different. A, B, C, and D. So depending on what species of bacteria you're looking at, they either use cytochrome A or cytochrome B or C or D if they do cellular respiration. let me stop there and ask if anybody has any questions. Hopefully this has been sort of a review for you for cellular respiration. I know the concept of the electron transport chain is very strange. It's just a very strange idea that there's this structure and these electrons are sort of fed into it, and they are literally passed through it until they come out the other end. And they are literally stepped down in energy as they pass through this protein chain. Their energy is taken from them, removed from them. And at the very end of the chain, there's a molecule there waiting to take them and be reduced. Remember, the energy that's harvested in the electron transport chain, that's going to be used to do some work. And that work is building a proton gradient, a hydrogen ion gradient. That energy is going to be used to fuel pumps in the cell membrane that's going to pump hydrogen ions from the cytoplasm into the periplasmic space. We're going to build a gradient. Gradients have stored energy in them. They have potential energy. So we took the energy from the electrons and we built a gradient that also has energy. And then we're going to let that energy dissipate. We're going to let the gradient resolve. The hydrogen ions are going to flow through the ATP synthase, and that energy will be used to build ATP. It's a long, complex way to make ATP, this process that we call oxidative phosphorylation, but it's extremely efficient. Remember, from one glucose, you end up with something like 30 ATP. So it's a great way to make ATP, even though it's very complex. So in anaerobes who do cellular respiration, most of them use inorganic molecules as their final electron acceptor. Now, remember what this word inorganic means in science. It means a molecule that does not contain carbon. Inorganic means no carbon. And remember, the top three, the ones you see most often as final electron acceptors, are nitrate, sulfate, and carbonate. Nitrate is the single most common final electron acceptor in anaerobic bacteria. Now, there are some bacteria that use organic molecules at the end of their electron transport chain. They're just not nearly as common. So there are some bacterial species that use something that contains carbon in it as their final electron acceptor. I've just written a few examples here. There's a molecule called fumarate. There's one called triethylamine. There's one called DMSO, or dimethyl sulfoxide. These can all be final electron acceptors too. They're just much less commonly seen. It's much, much more common to see these three, especially nitrate. The test that we're going to be doing today is going to look at this nitrate molecule as a final electron acceptor. In other words, we're going to look at a test that measures the ability of a bacterium to use nitrate as its final electron acceptor after glycolysis, pyruvate processing, Krebs cycle, electron transport. We're going to examine what happens at the end there. Does the nitrate get reduced? Oxygen gets reduced and becomes water. Nitrate gets reduced in these particular bacteria and becomes something else. Let's take a look at what nitrate becomes when it gets reduced, when it takes electrons at the end of the electron transport chain. This is the formula for nitrate here, NO3 minus nitrate plus electrons. Remember, it's receiving electrons from the electron transport chain. It's going to become a molecule called nitrite. That's NO2 minus. Nitrite and water will be created. Hey, that looks similar to what happens in aerobes, right? In an aerobe, oxygen receives electrons and a little bit of hydrogen and becomes water. Well, here the nitrate becomes nitrite with some water. Now, here's where it's different, very different from what happens with aerobes. Nitrite is a molecule that can be further reduced. Let me say that again because it's really important. Nitrate can be reduced to nitrite. But nitrite can actually accept more electrons. It can become further reduced if the organism has the enzymes to do that reaction. That's different from what happens in aerobes. There's only one reduction in aerobes at the end of the electron transport chain. Oxygen receives electrons and some hydrogen and becomes water. That's the only reduction that can happen in aerobes. But if you're an anaerobe and you use nitrate as your final electron acceptor, the reduction can sort of go on and on if you have the right enzymes. So in some bacteria that use nitrate, this is the final step. The nitrate becomes nitrite and we're done. But in other bacteria, they go on. They take the nitrite and they feed it even more electrons. And it becomes a molecule called nitrous oxide. And again, some bacteria stop there. But in some bacteria, the nitrous oxide can be even further reduced. It can also accept some electrons. And it becomes a molecule called nitrogen gas. Now that's as far as it can go. So some bacteria reduce nitrate to nitrite. Some bacteria reduce nitrate to nitrite to nitrous oxide. And some bacteria reduce nitrate to nitrite to nitrous oxide to nitrogen gas. It just depends. It depends on the species. So here's a nice test for not only examining microbes that do anaerobic cellular respiration, but for also characterizing how they do it in terms of what they do with their final electron acceptor. Remember, there are three very common final electron acceptor molecules in anaerobes: nitrate, sulfate, and carbonate. Nitrate is the most common. And because of the nature of nitrate, it can be reduced to different product molecules. It just depends on what enzymes the bacterial species has available. Now, just for completion's sake, bacteria that use sulfate at the end of their electron transport chain, sulfate will receive electrons, it gets reduced, and it becomes hydrogen sulfide and water. Microbes that use carbonate, that carbonate will receive electrons, again, become reduced, and create methane and water. So these equations are a lot simpler than what happens with nitrate, but nitrate's just more commonly used. So it's nitrate that we're actually going to look at today in the test. Does anybody have any questions so far? I know that's a lot of chemistry. Remember, for me, it's not important that you know the formula for these molecules. What's important is you know the names. You need to know the names, and you need to understand what's being oxidized versus what's being reduced and what those words mean. Something that's being oxidized is being broken apart. It's losing electrons. Something that's being reduced is gaining electrons. Any questions so far? All right, I'm going to jump back in. The first test that we're going to look at today is actually a very simple test. Remember when we talked about the Catalase test? We were examining to see if a microbe had the enzyme called catalase. And that enzyme, of course, breaks down hydrogen peroxide and creates oxygen gas. Well, this is a very simplistic test as well, this Oxidase test. It has one step. It's very easy to perform in the laboratory. What the Oxidase test is looking for is the presence of an enzyme, but this time that enzyme is called cytochrome C oxidase. It's looking for the presence of this enzyme, cytochrome C oxidase, in the electron transport chain. Remember, there are 4 kinds of cytochromes in bacteria: cytochrome A, B, C, and D. There's an enzyme associated with that cytochrome molecule. It's the enzyme that allows that cytochrome to accept electrons and then donate electrons. This enzyme is what allows the cytochrome to perform its job. And there are different versions of this enzyme. There's a version for cytochrome A. There's a version for cytochrome B, one for C, and one for D. We're looking at the one for C today. And that's what this test tests for. The enzyme is what allows cytochrome C to either gain electrons or lose electrons as it transfers them around the electron transport chain. Now, the way the test works is we grow some colonies on a plate and we expose them to a chemical called just the Oxidase reagent. All right, it's just a reagent chemical. You buy it commercially. When that reagent comes in contact with the cells, you're going to get a color change. It's going to turn purple if cytochrome C oxidase enzyme is present. So very simple test. Super simple test, okay? You buy this oxidase reagent commercially. You pick up some cells, pick up a colony off a plate. You can even do it right on the plate if you don't need the plate. You just put some of this reagent chemical, this oxidase reagent, on top of the cells. And if they have cytochrome C oxidase present, you're going to get a purple color change. Very simple. Now, one other thing to know, if you get a positive result, you know that that organism has cytochrome C oxidase in their cells. They use cytochrome C in their electron transport chain. Something else that means is that that type of bacteria also uses oxygen as their final electron acceptor. In other words, this test tells you that you've got an aerobe that uses cytochrome C in its electron transport chain, that version, and it has cytochrome C oxidase. If you use cytochrome C, you also have to have cytochrome C oxidase because those two things go together. But it's the aerobes that use this version of cytochromes, of the cytochrome. Okay, so we get some information from this test. The second test we're going to do is the one that's more complex. The second test that we're going to do is called the nitrate reduction test. And it's basically what we've just talked about. It's going to look at whether or not a microbe uses nitrate as their final electron acceptor, and specifically whether it takes the nitrate and reduces it to nitrite. All right, that was the first reaction we talked about. So what's going to happen is we're going to put the bacteria into this test broth. We're going to incubate it overnight. There's lots of nitrate in there. If that bacterium uses nitrate as its final electron acceptor, the nitrate in there is going to get reduced to nitrite. Now, this test uses a basic nutrient broth supplemented with nitrate. We just put a bunch of nitrate into a regular old nutrient broth. It's a potassium nitrate. That doesn't really matter. It's just a source of nitrate. Here's something that you should put a star next to, because this is kind of unusual. After we inoculate the medium, we're going to incubate it for 48 hours instead of 24 hours. That's kind of unusual. We don't usually do that. This has nothing to do with how fast or slow the cells grow, because you can do this test on any bacterium. So this is not about allowing enough time for the cells to grow, this 48-hour interval. Instead, it's about how much time it takes to do a full reduction of nitrate. Remember, some bacteria that use nitrate as their final electron acceptor, they're going to take that nitrate and they're going to reduce it all the way down to nitrogen gas. They're going to first convert it to nitrite. Then they're going to convert it to nitrous oxide. Then they're going to convert it to nitrogen gas. They're going to do 3 reductions. That takes time. That just takes time. So it's not that the cells need 48 hours to grow. No, I mean, some of the organisms we do this test on grow very fast. It's that the metabolic process takes time. So give it a full 48 hours in the incubator. Now, when you take the tubes out of the incubator, you have to do the first of several steps. This is where this test gets complex. The first thing you do when you take the tube out is you add 2 reagents to it. Now, it's not important that we know the names of these reagents. Most test kits, because you usually buy your nitrogen reduction tests, you buy them commercially. It's just easier than making them yourself. They come with 2 reagents that are usually labeled reagent A and reagent B. So that's what everybody refers to them as. You're going to drip those 2 reagents into the tube. Now, if the nitrate in the broth has been reduced to nitrite, the tube's going to turn red. That's the first thing to know about this test. If you take the tube out of the incubator, you put in reagent A and reagent B. If the broth turns red, you've got a positive test. That tells you that the nitrate that was inside the broth was reduced to nitrite. That bacterium, whatever it happens to be, uses nitrate at the end of its electron transport chain. And as it gains electrons, it becomes nitrite. That's what this test looks for. Looks for that nitrite molecule. If it's in there, when you add A and B, you're going to get a red color. Okay, positive test. If you take the tube out of the incubator, though, and you put A and B in, and there's no red color, you're not done. You're not done. Because remember, there are some bacteria that can further reduce nitrite. So if you take the tube out of the incubator and you put A and B in, and you get no color change, no red color, you have to do the second step, which is add some zinc. We put some zinc reagent into the tube. Now, what does zinc do? Zinc is a metal that can force the reduction of nitrate to nitrite. In other words, if there's no red color in that tube, it may be that the bacterium doesn't use nitrate. It may be that that particular bacterium doesn't use nitrate at the end of its electron transport chain. Maybe it uses sulfate. Maybe it uses carbonate. Who knows? If there's no red color, we put zinc in. If there's any nitrate in the tube, it's going to be reduced to nitrite by the zinc. The zinc is doing the reducing. It's not that the bacterium reduced it. The bacterium couldn't. It couldn't do it. That's why there was still nitrate in the tube. When we put the zinc in, the zinc will reduce the nitrate to nitrite, and you'll get that red color change because now there's nitrite in the tube. But here's the important part. That's a negative test. Now you might say, hold on a minute. How can I have red color and it be a negative? The reason it's negative is because the bacterium didn't reduce the nitrate. The zinc did. The zinc did. By adding the zinc, if we get a red color, we know that there must have been some nitrate in that tube because zinc will reduce nitrate to nitrite, and that will set off the color change. The bacterium didn't do the reduction, though. The bacterium didn't do it. The zinc did. That's a negative test. That tells us that the bacterium was not able to reduce nitrate. The zinc did it. Okay, are you still with me? We're not done yet. I told you this was a complex test. Let's say you put the zinc in the tube and you still don't get a red color. What does that mean? Well, that means that the bacterium must have further reduced the nitrate. In other words, it took the nitrate, it reduced it to nitrite, but then it further reduced it either to nitrous oxide or to nitrogen gas. Some bacteria can do that. Some bacteria can further reduce nitrate past nitrite. So there's no nitrite in the tube anymore because it's been further reduced to either nitrous oxide or nitrogen gas. So how do we know the difference? How do we know which was the final product? Was it nitrous oxide, or was it nitrogen gas? We use our old friend, the Durham tube. Remember the little tube? You put it upside down in the nitrate reduction test tube, and you look for a gas bubble. Nitrous oxide will just dissolve in the medium. But if nitrogen gas was produced, it'll get trapped in the Durham tube. Candy's asking a great question. So if you have a red color with the second step, do you still have to do the third step? That's a great question. In other words, you do the first step, you add A and B, and it doesn't turn red. So you add zinc and it does turn red. Technically speaking, Candy, you've got a negative right there. You've already got your negative, so you don't need to do the third step. I'll tell you what, though, most microbiologists, I'll say most scientists, will tell you you should always do the full test. You should always do the full test just to be complete. But you're right. At that point, you already know what you need to know, and that's that this organism can't reduce nitrate to nitrate. It can't do it. Okay. And the reason we know that is because there must have been nitrate left in the tube. Otherwise, it would not have turned red. Zinc reduces nitrate to nitrite. That's a great question, Candy. Good thinking. So here's a summary for this nitrate reduction test. We're going to take our organism and we're going to inoculate nitrate reduction broth. Remember, this broth is basically a nutrient broth, a growth medium, and it's been supplemented with a bunch of nitrate. What this test is really looking for, what this test is able to tell us, is whether or not the bacterium can take that nitrate and turn it to nitrite, because nitrite causes a color change. We take the tube, we inoculate it, we put it in the incubator for 48 hours. And when we take it out, we put our first two reagents in. We put in reagents A and B. If the bacteria used nitrate at the bottom of its electron transport chain, and the nitrate accepted electrons and became nitrite, that nitrite will react with reagents A and B, and you'll get a red color in the tube. That's a positive reaction because the reagents are telling us that there's nitrite present in the tube. Red color there means nitrite is present. And the only way we could get nitrite is if the organism reduced the nitrate. All right. Now, if no red color develops after A and B are added, there are two possible explanations. Either nitrate is still in the tube because the organism doesn't use nitrate at the bottom of the electron transport chain. It doesn't use it, so it didn't reduce it. That's one explanation. The second explanation is that the organism does use nitrate, but it reduces it past nitrite. Maybe it took the nitrate, converted it to nitrite, and then further reduced it either to nitrous oxide or to nitrogen gas. Neither nitrous oxide nor nitrogen gas is going to turn that tube red. Only nitrite does that. So how do we know if we have nitrous oxide in there or nitrogen gas in there? The tube has no color in it. How do we know which is which? Well, we're going to put a Durham tube in there. If nitrogen gas gets produced, it'll get trapped in the Durham tube. So we have two possible things going on. Either the organism doesn't use nitrate as its final electron acceptor, or it does use it, but it reduces it beyond nitrite. And we have to be able to tell the difference between these two. So the second step is to add zinc. If the organism doesn't use nitrate, if the first explanation is correct, then that nitrate is still sitting in that tube. When we add zinc to it, The zinc will reduce it to nitrite. Now you have nitrite in the tube, you're going to get a red color change. That's a negative test result, because that told us that the microbe did not reduce the nitrate. The zinc did. If you add the zinc reagent and you get no color change, well, now you know you've got a microbe that uses nitrate and reduces it somewhere beyond nitrite. It's either turned the nitrite into nitrous oxide, or it turned the nitrite into nitrous oxide and then nitrogen gas. But either way, you've got a positive test, because this organism was able to reduce nitrate. So either way, whether it took the nitrate to nitrous oxide, or it took the nitrate and made it into nitrogen gas, either way, you've got a positive test. So how do we know what happened? Because I've got no red color, I've got no indicator in there. It still looks like it started. It's a positive test, but I didn't get a red color change. How do we know whether we've got nitrous oxide in there or nitrogen gas in there? The answer is we look in the Durham tube. If there's a gas bubble in there, we know it's nitrogen gas. Nitrous oxide actually dissolves into the medium, but nitrogen gas doesn't. So nitrogen gas will be trapped as a bubble in the Durham tube. So if you see a bubble, the bacteria reduced the nitrate all the way to nitrogen gas. If you don't see a bubble, the bacterium reduced the nitrate to nitrous oxide. What a handful of results. This is a complex test. This is a very complex test, but it's useful because not only do we know whether or not we've got an anaerobe doing cellular respiration, we know that it's using or not using nitrate as its final electron acceptor. And we also know what end product is produced, because that's another characteristic, another piece of data that we can use when we're trying to identify these microbes. So we're going to run these two tests. These are the organisms that we're going to use today. We're going to use a new organism called Streptococcus Pneumoniae. Gosh, anytime you see a species name Pneumoniae, you know you've got a troublesome organism. Remember, we looked at Klebsiella pneumoniae, and we said sometimes that can be a really bad pathogen. Streptococcus pneumoniae is another really bad pathogen. These Pneumoniae species, they cause what we call pneumococcal diseases. A Streptococcus pneumoniae causes pneumococcal disease. And there are lots of different versions of pneumococcal disease. There's pneumonia, that's a big one. You can also get meningitis from strep pneumoniae. You can get septicemia, that's a blood infection. There are hundreds and hundreds of strains of Streptococcus pneumoniae. There are hundreds of different strains. Most of them are pathogens. This is a bad bug. And that's why we have a vaccine available against this particular bacterium. Now, this is a vaccine we don't give to children. We don't give it to children because children don't tend to be susceptible to these pneumococcal diseases. Children don't tend to get infected with strep pneumoniae nearly as often as older adults do. So by using epidemiological data, we know that it's the older part of the population that needs to be vaccinated, and we vaccinate them. It kind of varies. The recommendations change periodically, but generally people over the age of 50 are the ones that are getting vaccinated against Streptococcus pneumoniae. And again, we call it the pneumococcal vaccine because it will protect you against any pneumococcal disease, pneumonia, meningitis, septicemia. So it's people in my age group that are particularly susceptible to strep pneumoniae, and we're the ones who get the vaccine. So we'll take a look at that organism. Because it's a streptococcus, of course, it's gram-positive. It's a gram-positive coccus. It tends to be found in the respiratory tract. So pneumonia is one of the primary diseases it causes. But because it's so pathogenic, because it's so virulent, it can also then spread into the central nervous system and into the bloodstream. There aren't a lot of bacteria that are capable of this. This tells you you've got a really bad organism. You've got a really pathogenic organism. The second bacterium we'll use today is our old friend Staphylococcus Epidermidis. You've seen this one before. This is another gram-positive coccus, but this one has a lot of strains that are not pathogenic. Remember, you find Staph epidermidis on the skin on humans, but sometimes there are pathogenic strains. We'll use our friend E. coli, gram-negative rod, lives in the intestine, coliform. And we'll also use Pseudomonas aeruginosa, gram-negative rod. Remember, this one lives out in the world. It likes wet environments. It tends to hang around in hospitals and so on, where the facility stays damp. It's also an opportunistic pathogen, remember. So 4 organisms, 2 of them are gram-positive, 2 of them are gram-negative. Let's do some tests. This is a good time for us to take a short break. Right now, I have about 10:02, so we'll come back at about 10:07, okay? Take a break, get up, stretch your legs, grab another Monster or Red Bull or coffee or tea or whatever it is. Pick your poison, and I'll see you back in a few minutes. Okay. All right. Go ahead and type your name in chat for me again. Thank you. And we will take our bacteria and we'll do some testing. Yeah, as you come back in, just type your name into chat for me. That's my record that you attended lab today and that, you came back after the break. You attended the whole lab. So yeah, go ahead and type your name in for me. All right, so let's take a look. Let's do our oxidase test first. Oh, before I do that, I forgot I had put some imagery in here for you. This is a picture. I actually got this off of the CDC website. This is the Strep pneumoniae organism under the microscope. And this was in a patient who died of septicemia. So what you're looking at here, this was a blood sample that was taken out of the patient and put onto a microscope slide. We created a smear. Now, this doesn't look like blood. You don't see these nice red blood cells in here. You don't see a bunch of white blood cells or anything. It doesn't look like a normal blood sample. This patient was very, very sick. And by the time the sample was taken and put on the slide, you got some artifacts created. This pink material, there's a lot of, there are red blood cells trapped in here. There's a lot of fibrin in here, a lot of inflammatory material in here. That's why it looks so abnormal. You can see this sort of web-like, this is all fibrin. So it doesn't look like a normal blood smear at all. But what I want you to look at is the dark things in here. This is the Streptococcus organism. Now we're under the 40x lens here, but you can still see a lot of detail. If you look up here on the upper left, there are 3 cocci in a row right there. So you can see 3 individual cells up here. There are other places where there are 2 cells in a row. Here's another one right here. Here's another doublet and another doublet. Streptococcus, remember, is an organism that likes to form chains. Here's another chain of 3, chain of 2. Here's a nice long chain right there. So this is an organism, again, that has hundreds of known strains. And the problem is that almost all of those strains are pathogenic. Very, very bad organism. This image right here is taken with a fluorescent microscope. Now, again, you can see the streptococci in here. You don't see any of the other material on the slide, and that's because of the microscope that was used. What they did here was they tagged the streptococcus cells with an antibody. So an antibody that's directed against these organisms that has a fluorescent molecule attached to it. So anywhere that you see this bright white color, you know you've got a streptococcus. You can imagine that using these fluorescent antibody tags is a great way to examine a specimen for a particular microbe. The antibody will only stick to that microbe, so you'll know if it's present. And the antibody has a molecule on it that fluoresces underneath that microscope. So you basically remove everything else from the image, and all you're left with is the microbe if it's present. So yeah, this person was also infected with Streptococcus pneumoniae. This sample was actually CSF, cerebrospinal fluid. So this person had meningitis. Over here, I hate to show you something gross so early in the morning, but this is a brain that was taken at necropsy sorry, I always say necropsy because I'm a veterinarian. It was taken at autopsy from a person who died of a streptococcal meningitis, a pneumococcal meningitis. What you're looking at here is the ventral surface, the bottom surface of the brain. Where you have normal brain tissue, it's this sort of a brownish red color. Right here, you can see a lot of white color. I know, hold on. This is pus. This is pus. This is what we call a superrative meningitis. There's so much bacteria in here, and the immune system is attacking it. So everywhere you see white, you've got bacteria, you've got white blood cells in the area, furiously trying to kill it. But this patient did not survive. Pneumococcal disease is a bad disease. Okay, enough of gross things for one morning. Okay. So the oxidase test, you can do it a couple of ways. One really common way to do it is to grow the bacterium on an agar plate. You can see what they've got here is a quadrant streak. You can see the streaks in the 4 quadrants. We've got individual colonies here in the 4th quadrant. You can actually put the reagent right on the plate. You can just drip it right on the plate if you don't need this plate for something else. And if a purple color develops, it tells you that the cells are making cytochrome C oxidase. Another thing you can do, let's say you wanted to use this plate for other tests and you didn't want to ruin a portion of it with the reagent. You could take a sterile cotton swab. You can pick up one colony off of the plate on your cotton swab and then drip the reagent right down on the cotton swab. If it's positive, you're gonna get a purple color. So this is a positive result. This is a negative result. We read Oxidase test results immediately, just like we did with the Catalase test. Remember, with Catalase, we took some cells, we put them onto a microscope slide, and we put some hydrogen peroxide on top. We put a drop of hydrogen peroxide on the cells, and we looked for bubbles, oxygen bubbles. That told us that those cells made catalase, and we're breaking down the hydrogen peroxide into oxygen and water. What's happening here is the oxidase reagent is reacting with cytochrome C oxidase and creating that purple color. So you know you've got that enzyme present. If cytochrome C oxidase is present, we know that that organism uses cytochrome C in its electron transport chain. We also know that it performs aerobic cellular respiration. Now, something else that's important to know about the oxidase test, and it relates to this picture over here. You can buy this oxidase test reagent. You can buy it in bottles. They're small bottles. They have like 5 to 10 mLs of reagent in them. Relatively, it's expensive to buy a whole bottle of the reagent. You only need a drop at a time, though, for a test. So if you were doing a lot of oxidase testing in your lab, it might be worth buying a bottle of the reagent. But here's the problem. Once you open the bottle of oxidase reagent, it starts to degrade. As soon as that reagent gets exposed to the air, it starts to degrade. So even if you're really careful and you open the bottle and you squirt one drop and then you close the bottle again, it's too late. It's already starting to degrade. So they don't have a very, this bottle of oxidase reagent doesn't have a very long shelf life. It's months. You don't get even a year out of a bottle. It's something like 6 months, and it's bad. So unless the lab is doing a lot of oxidase testing, sometimes they don't want to buy a bottle because it'll go bad before they have a chance to use it all. Luckily, there's another option. If you're not doing that much oxidase testing in the lab, you can purchase what are called Oxidase Test strips. And that's what you can see on this slide. You can buy a box of these strips. And what these strips of paper are, they've been impregnated with the reagent. So it's a slip of paper that you could hold between 2 fingers. And it's been impregnated with the oxidase reagent. So all you have to do is you, again, pick up some cells with a sterile swab and just smear them somewhere on the strip. And what'll happen is, again, it'll turn purple if cytochrome C oxidase is present. It won't turn purple if it's negative. So again, if you're a lab, if you're a small lab, or you're a lab that just doesn't do that much oxidase testing, it's much smarter, it's much more economical to buy the strips. Each strip is packaged in a sterile paper package, and it lasts much longer. So a box of strips can last, you know, a couple years without going bad, without expiring. Bigger labs, you know, they'll buy the reagent in a bottle because they go through it fast. So it's not a problem. But the smaller labs, they tend to skip the bottle, buy the test strips because they'll last longer. So what we did was we took our 4 organisms, our 2 gram positives and our 2 gram negatives, and we made a quadrant streak plate for each organism. Then I took a colony off of each plate with a sterile cotton swab, and I put a drop of oxidase reagent on top, and I looked for a purple color change. And here are the results. Streptococcus Pneumoniae, negative, no purple color change. Staphylococcus Epidermidis, negative. E. coli, negative. Pseudomonas aeruginosa, positive. So I only got 1 positive here. Pseudomonas aeruginosa. So let's think about how we would interpret this. Pseudomonas aeruginosa tested positive for cytochrome C oxidase. Cytochrome C oxidase is the enzyme that allows cytochrome C to do its job in the electron transport chain during cellular respiration. And its job is to help the electrons pass down the electron transport chain. So cytochrome C is the molecule that's helping the electrons. Cytochrome C oxidase is the enzyme that's allowing cytochrome C to do its job. We can also conclude that Pseudomonas aeruginosa is doing aerobic cellular respiration because it uses the C version of cytochrome. It uses cytochrome C. Make sense? What about those negative results? What can we conclude about those? 3 of our organisms were negative on the Oxidase test. So they do not have cytochrome C oxidase. That means they do not use cytochrome C in their electron transport chain. Can I conclude then that they do not do aerobic cellular respiration? We said that organisms that have cytochrome C in their electron transport chain, we can conclude that those use aerobic cellular respiration. If they don't have cytochrome C, can I say, oh, maybe they don't do cellular respiration? Can I go that far with my conclusion? What did we say about cytochromes? How much of a conclusion can I draw? Those 3 organisms that were negative on the oxidase test, they were negative for the presence of cytochrome C oxidase. If they don't have cytochrome C oxidase, they don't have cytochrome C, because that's what that enzyme works with. Does that mean they don't do cellular respiration? Because if you do cellular respiration, you have to use cytochromes. So if they don't have cytochrome C, does that mean they don't do cellular respiration? What do you think? What, how might you explain that? Candy's saying, no, that does not mean that they're not doing cellular respiration. If they were doing cellular respiration, how come they don't have any cytochrome C? You need cytochromes to do electron transport. They don't have any cytochrome C. So how could they possibly be doing electron transport? How could they possibly be doing cellular respiration? Type it into chat for me. What did we say about these cytochromes in bacteria? If they don't have cytochrome C, does that mean they don't have any cytochromes? You have to have cytochromes to do electron transport. And you have to do electron transport to do cellular respiration. So if you don't have cytochrome C, does that mean you can't do cellular respiration? Candy's got it. Candy's got the answer. Good job, Candy. No, it doesn't mean that. No, they could be using a different cytochrome. There are 4 families of cytochromes. Bacteria can use cytochrome A, they can use cytochrome B, they can use cytochrome C, and they can use cytochrome D. So all the oxidase test tells us is that they're not using C. We would need more tests to know what kind of cytochrome they were using. Okay, all the oxidase test tells us is whether or not they're using cytochrome C. Okay, that's important to note. You cannot say that an organism that's negative on the oxidase test is not doing cellular respiration. You can't say that. They're just not using cytochrome C to do it. They could be using a different kind. But good old Pseudomonas is using it. That's an interesting thing to know. All right. Next test is the nitrate reduction test. I took some nitrate reduction test broth, which remember is just nutrient medium that's been supplemented with nitrate. I inoculated 4 tubes, one with each of our organisms. I put those tubes into the incubator and I left them there for 48 hours. And then I pulled them out of the incubator. Okay. 37C, 48 hours. pull them out of the incubator, you have to add your first reagents. And remember, the first reagents we add are reagents A and B. They've got the names listed on this slide. You don't need to know these names. Just know that 2 reagents are added, and we tend to call them reagents A and B. Now, on this flow chart, we have 2 possible results. We're either going to see a red color or we're going to see no color change. If you see a red color, that means that you've got a positive test for nitrate reduction. After you add A and B, if the tube turns red, you know that the organism took the nitrate and reduced it to nitrite. It's the nitrite that causes the red color change. If the tube stays colorless, though, you can't stop. Okay, you can't stop because 2 things are possible at that point. Either the bacterium doesn't use nitrate, or it reduces nitrate beyond nitrite. So the next thing we have to do is add zinc. If you get a red color, you've got a negative result. Red color means that right here, this colorless tube still had a bunch of nitrate in it. And when I added the zinc reduced the nitrate to nitrite. And the nitrite gave me the red color. It's a negative test because the bacterium didn't reduce the nitrate, the zinc did. The other thing that might happen is I might still stay as a colorless tube. If it's colorless after the zinc is added, I know I've got a positive test. What that means is that there was no nitrate in this tube. Because if there was nitrate in there, the zinc would have reduced it. So instead, this colorless tube means that the bacterium reduced the nitrate to nitrite and then to some other chemical, either nitrous oxide or nitrogen gas. It's a positive test for nitrogen reduction. How do I know which product was made in this colorless tube? Well, I'd have to look in the Durham tube. If the Durham tube has a little gas bubble in it, I know that the bacterium reduced the nitrate to nitrite, and then to nitrous oxide, and then to nitrogen gas. If there's no gas bubble in the tube, I know it reduced the nitrate to nitrite, and then to nitrous oxide. Positive test at this point. This medium starts off like so many of our media do. It has this just boring beige color. This is what we mean by a colorless result. No color change. This is that red color that we look for. Okay. Here are our results. In this tube over here, this tube had Streptococcus Pneumoniae in it. You can just barely see the Durham tube here. I know it's hard to see, but I'll tell you what was actually in the Durham tube, if anything. This is the Strep Pneumoniae tube after zinc was added. After zinc was added, the tube turned red. So think about what that means. These 2 tubes, Staph Epidermidis and E. coli, these turned red after reagents A and B were added. Now I know it says N1 and N2 here, but it's the first 2 reagents, A and B. A and B were added, tube turned red. Pseudomonas aeruginosa, this is what that one looks like after the zinc was added. And again, I know it's hard to see what's up here, but I can tell you that in this tube, there's a gas bubble in the derm tube. So let's think about this. Streptococcus pneumoniae tube turned red after the zinc was added. Positive or negative for nitrate reduction? Turned red after the zinc was added. Positive or negative? First tube, red after zinc added. Positive or negative? Type it into chat for me. You got a 50% chance, right? You got a 50% chance here. So we're not given all the information, are we? We're just given the last piece of information, that after we added zinc to it, it turned red. Type it into chat for me. I took that Streptococcus pneumoniae tube. I took it out of the incubator. I added reagents A and B to it. No red color change happened. So I added the zinc and it turned red. What does that mean? Is it positive or negative for nitrate reduction? What do you think? Candy's got it. If the tube turns red after zinc is added, it's a negative result. Think about what the red color is telling you. The red color means that that tube has nitrite in it. That's what the red color means. The reason it's a negative result is because the bacterium didn't reduce the nitrate to nitrite. The zinc did. The bacterium must have left the nitrate alone. The nitrate must have still been in the tube when I added the zinc, because zinc will take nitrate and reduce it to nitrite. And nitrite makes the tube red. The bacterium didn't reduce the nitrate. The zinc did. Negative, negative. Good job, Candy. Second set of tubes. These two organisms came out of the incubator. I put A and B in, and they turned red. They turned red after A and B were added. I didn't even add zinc. What does that mean? Positive or negative for nitrate reduction? You take the tubes out of the incubator after 48 hours, you add reagents A and B, and it turns red. What does that mean? Is that positive or negative? Remember what red means. Red means there's nitrite in the tube. Candy's got it again. Candy's on fire today. Those two organisms are positive for nitrate reduction. The organism itself took the nitrate and reduced it to nitrite. And the nitrite turned the tube red. Because it turned red after A and B were added, we know we have a positive result for nitrate reduction. The Pseudomonas is the only really different one. The Pseudomonas remained colorless. We added A and B, it stayed colorless. We added zinc, it stayed colorless. Remember, if it's colorless after the zinc is added, you've got a positive test. It seems so strange. The reason it's positive is because the bacterium reduced the nitrate It did, but it took it beyond nitrite. If it took it to nitrite, the tube would have turned red. It didn't turn red. So that tells us it must have then taken the nitrite and reduced it even further to nitrous oxide, perhaps, or perhaps to nitrogen gas. And the only way we would know is to look in the Durham tube, which one of those two was produced. And if you look in this Durham tube, and again, I know it's hard, but if you look in this Durham tube, there's a gas bubble in here. You can just see the top of the liquid line, and then there's gas above that. So Pseudomonas aeruginosa is positive for nitrate reduction, but Pseudomonas can reduce it all the way to nitrogen gas. Pseudomonas is positive, Staphylococcus Epidermidis is positive, E. coli is positive. Our only negative was the Strep Pneumoniae. One more little thing to note here. This is one of those sort of sidebars to note. If you take this nitrogen reduction tube out of the incubator, and you can already see a gas bubble in the Durham tube before you even add A and B, you know you've got a positive result. Let me say that again and see if you can figure it out. If you take that nitrogen reduction test tube, if you take that broth out of the incubator, before you even add A and B, you notice that there's a gas bubble in the Durham tube. You know you have a positive result. You have a positive result, and the organism reduced the nitrate all the way to nitrogen gas. That's the only way that there can be a bubble in the Durham tube. So that's one of those times when the technician who's doing the work, if they understand this test, they can sort of have a little shortcut there. The only way there's going to be a gas bubble in the Durham tube in a nitrogen reduction test is if the organism is positive for nitrate reduction and is capable of reducing nitrate all the way to nitrogen gas. Now, I'll tell you, in the labs that I've worked in, we would never actually stop there. Even though we know we've got a positive, we would confirm that by finishing the test. But yeah, if you spot that gas bubble right away when you take the tube out of the incubator, you've got a positive result. But like I said, for completeness sake, to confirm that, we would still add A and B, and we would still add the zinc, and we would see no color change. And then we would note, well, there's a gas bubble in there. So just a little extra thing, a little extra trick in the laboratory, because we love little tricks in the laboratory. All right. Any questions about this? Any questions about this very complex test? Any questions about what it means? Remember, this is all about nitrate reduction. And the reason a bacterium would reduce nitrate is because it's using nitrate as its final electron acceptor. And that means it's performing anaerobic cellular respiration to make its ATP. Those are the only organisms that are reducing nitrate, the ones that are doing anaerobic cellular respiration. If you did a nitrate reduction test and you got a negative, if you did a nitrate reduction test and you got a negative result, remember that would mean it was red after you added the zinc. You cannot conclude that this is not an anaerobic cellular respiration organism. You can't conclude that. It could be that it's using a different electron acceptor. Maybe it's using sulfate. Maybe it's using carbonate. All we can say is it's not an anaerobic cellular respiration process with nitrate as the final electron acceptor. That's all you can conclude there, okay? Microbes are very diverse in their metabolic abilities. We constantly have to remind ourselves of all the variations that they can perform. There are different ways to do anaerobic cellular respiration. There are different final electron acceptor molecules. Most of the time, if you're doing anaerobic, it means that you're using nitrate. Most of the organisms that do anaerobic cellular respiration use nitrate, but some use sulfate, some use carbonate, some use some of those other molecules I mentioned, fumarate or triethylamine or DMSO. The variety is endless. So it's very important that we know how to interpret the results that we get, so we can accurately conclude what kind of an organism we're dealing with. All right. We have a few minutes left. And before I let you go, I just wanted to draw your attention to one more thing on our Canvas page. So again, we're here in week 9. Next week is week 10. That's the last week of class. And then we have our final exam week in week 11. The week of August 1st is our final exam week. You do have a to-do list for the final exam week. It just reminds you what the final exam is going to be about. Okay, so I just wanted to quickly go through this and show you a study document that I posted for you. The final exam is going to have 40 questions on it, and it's going to be worth 100 points total. Okay. Now, the exam is split into 2 parts. The first part is about previous lecture topics. So stuff you've already been tested on, review material. About 20 questions are going to be review. The other 20 questions will be from the new material that you've been learning. So there'll be questions from past lecture material, about 20 of them. And then there'll be 20 questions about new material. And the new material includes the immune system, vaccination, epidemiology, and pathogens and how they spread. Now, here's how I do the review material, because I don't think you have the time to adequately review all of the past lecture material. The way I do it is I put a document together for you that contains a bunch of questions on it, exam questions, basically. There are about 50 questions on that document. And all of those questions are about previous lecture material. I'm going to pull that document up in just a moment. But what you're going to see is that there are questions and answers. So to study the old material for the final, this is the document you want to study. If you learn the answers to all 50 of those questions, you'll be ready for the test, because I'm going to draw 20 of those questions and I'm going to put them on the final. That's for the review material. You'll still have to learn the new material for the other half of the exam. But for the review material, you'll just have to learn the correct answers to the questions that I've provided for you. Okay. The exam opens up on Monday morning, August 1st, and is due by midnight on August 3rd, Wednesday, August 3rd. Please keep this in mind. No late submissions will be accepted for the final exam. Okay. We're at the last moment of the course. I have to get the exams graded and into the registrar, the grades into the registrar. So I cannot accept late submissions on the final. Now, of course, if an emergency happens, of course, I'm going to work with you. But you cannot just submit the final late. Okay, that cannot happen this time. So unless an emergency happens, you've got to get the final into me by Wednesday, August 3rd by midnight. Okay. Now let me show you this review document. Here's that final module, the week of August 1st. Here's where you'll be taking the final lecture exam. Half of that exam is on new material, half of it's on old material. And here's how you're going to study the old material. You're going to click on this, review material questions to study for the final exam. This is a list of about 50 questions. All right. And you'll notice I've got the question and I've got the answer indicated for you as well, the correct answer. So what you need to do is learn the correct answers to these questions. There's 50, I guess it's 49 questions on here. Yeah, 49 questions. I'm going to choose 20 of them. I'm going to choose 20 of these review questions and put it on the final. So if you learn the answers to all of these questions, you will get all of the review questions right on the final. Okay. Which of the following is the definition of the term microbe? A single-celled microscopic organism or acellular particle. The three domains of living things are bacteria, archaea, and eukarya. Okay, hopefully that'll make your studying much easier. Learn the answers to those questions. I'm going to pull 20 of them and put them on the final. And that'll be the review portion. And the rest of the questions will come from the new material. Okay. Are there any questions that I can answer right now? Either about what we've talked about today or about the final exam? Any questions? All right, very good. Thank you very much, everybody. I'm going to let you go. Enjoy the rest of the day. Stay cool. It's going to be very warm and we're under a heat emergency. Hydrate. Don't overdo it outside. I sound like your mother, don't I? Don't overdo it. Stay in the AC if you can. Hydrate. If you haven't finished lecture exam 4, be sure to get that to me by midnight. And I will see you on Monday. All right, everybody.